Okay, here we are looking at infradian rhythms. Infradian rhythms, these are biological rhythms that have a frequency of less than 1 in 24 hours. In other words, it takes them longer than a day to complete. They last longer than 24 hours. We're going to be looking at two key examples. We're going to be looking at the female menstrual cycle, and we'll also be looking at seasonal affective disorder, or SAD. Okay, so let's start with the female menstrual cycle. I will show you this video in class. This is an example of an infradian rhythm that is governed by monthly changes in the hormone levels that regulate ovulation in women. The cycle refers to the time between the first day of a woman's period to the day before her next period, when the womb lining is shed. The cycle takes about 24 days to complete, although anywhere between sort of 24 and 35 days is generally considered normal. During each cycle, you have rising levels of the hormone estrogen. This causes the ovary to develop an egg and release it in what we call ovulation. If the woman becomes pregnant, obviously, then that egg will stay in the womb. If not, then the egg is absorbed back into the body. Throughout the cycle, after ovulation, we then have rising progesterone levels. This helps the womb lining to grow thicker and gets the womb ready for pregnancy. If pregnancy doesn't occur, then the egg is absorbed into the body, the womb lining comes away and leaves the body in menstrual flow, and this is menstruation. So we can see here with this cycle that we've got a few different phases, and this one, it shows a 28 cycle, and generally that's what you will see, but really it varies so much from woman to woman. We have follicular phase, we have the ovulation phase, luteal, premenstrual, menstrual phases, but you don't need to know the exact terms. As long as you can describe generally what's happening with the hormone levels throughout the month and what that is associated with in terms of ovulation and menstruation. Okay, so now bear with me because this is quite an interesting study, but I need you to know in advance that it fails replication. So everything you hear, take it just with a real pinch of salt. Okay, so the menstrual cycle is an endogenous system. There is no doubt about that. But there is evidence that it may be influenced by exogenous factors. Here we're going to be looking at other women as those in exogenous factors, but other things like stress and diet, exercise, even male sweat containing testosterone can influence the female menstrual cycle. So let's take a look at this research by McClintock. This is a really famous study, I guess maybe now almost infamous. She found that female menstrual cycles can synchronize as a result of female pheromones. Now, even that is questionable, but we continue. They studied 29 women that had a history of irregular periods. Samples of pheromones, I'm going to put that in inverted commas. Samples of pheromones were gathered from 9 of the women at different stages in their menstrual cycle. They had cotton pads that were placed under their armpits and worn for at least 8 hours to ensure that those pheromones, inverted commas, were picked up. The pads were then treated with alcohol and frozen to be rubbed on the upper lip of other women. On day 1, they rubbed the pads from the start of the menstrual cycle to the women. On day 2, they were all given the pad from the second day of the cycle, and so on and so forth. McClintock found that 68% of women experienced changes to their cycle, bringing them closer or more in line to their odor donor. Yes, that really happened. This was called the McClintock effect after the researcher, and it was published in the late 90s in Nature, the premier journal for scientific research. So it was a huge, huge study at the time. But we will look at how this has since been clearly, obviously debunked. All right, let's have a look at another infradian rhythm now, one that you may have heard of, which is seasonal affective disorder, or SAD disorder. It's a depressive disorder. It has a seasonal pattern of onset, and it's described in the DSM. As with other forms of depression, we typically see persistent low mood alongside general lack of activity and interest in life, etc. It is colloquially termed the winter blues. This is because the symptoms are triggered during the winter months when the daylight hours become shorter. But it is absolutely far more than just feeling a little bit sad. It is a specific type of infradian rhythm known as a circannual rhythm, because it has that yearly or annual cycle. It is hypothesized that the hormone melatonin is implicated in the cause of SAD. During the nighttime, the pineal gland secretes melatonin until dawn, when there is an increase in light. During winter, the lack of light in the early morning means that the secretion of melatonin continues for longer. And this is then said to have a knock-on effect on the production of serotonin in the brain. And this, as we know, is a chemical that is related to depression. So the fact that nighttime is lasting longer means that the secretion of melatonin is lasting longer, which means that the production of serotonin is becoming inhibited. And this is the current hypothesis and the current thinking about why SAD occurs. All right, so let's move on to evaluating our infradian rhythms. So our key support for the menstrual cycle and the role of exogenous effects on the menstrual cycle comes from that McClintock study. There were arguments at the time, there were discussions. The field of evolutionary psychology was really coming to prominence at that time. And there were evolutionary explanations as to this synchronization effect. They thought it had evolutionary value. Basically, the prevalent theory was that it was an evolved strategy among females to encourage cooperation. Basically, to stop becoming some kind of harem for a single dominant man. The idea was that if women had synchronized cycles, they'd all be fertile at the same time, so one man would not be able to reproduce with all of them. It was kind of a protective strategy. If females were all sexually receptive at the same time, then it would be really difficult for a male to control the sexual access to particular females at all times. So it was thought to be sort of a cooperative strategy. Other evolutionary theories was the idea that if women were all menstruating together, then they'd all become pregnant, they'd all ovulate at the same time and become pregnant at the same time. So in social groups, this means that really we become then part of a social group where, you know, if a baby had lost their mother during childbirth, for example, didn't have access to breast milk, then there would be other women who were breastfeeding their own children who would be able to take over that role, and therefore it would be a survival strategy. Now, these theories are absolutely rubbish. They're post hoc. They're not based on any evidence. They're just speculation. So please don't take them seriously as any sort of scientific theory. But that's kind of the idea behind McClintock's research there in terms of synchronization of the menstrual cycle. So let's then take a look at all of the issues with McClintock's research into synchronization. You can read about this online. It has been totally debunked. There's a lot of criticism of her study for a whole variety of reasons. First up, the idea that there are so many confounding variables. There are so many other factors that influence a woman's menstrual cycle. As we said before, stress, diet, exercise, male sweat, and therefore testosterone all influence the menstrual cycle. So these may act as confounding variables, which means that the supposed pattern of synchronization is no more than what would be expected to occur by chance. There are other issues in terms of the small samples, and there's issues regarding the selection of the participants as well. There seems to have been some sampling bias there. Just as a little interesting side note, McClintock studied at the same college as Hillary Clinton at the same time, and all of the participants came from McClintock's college. So there is rumour that one of these women who had sweat put on her upper lip was indeed Hillary Clinton. So that's one issue, is that there is perhaps sampling bias in this research. There was also poor statistical analysis. There was failure to control adequately for the convergence of menstrual cycles that would occur just by chance. So let's just take a look at this. Given that the average menstrual cycle length is 28 days, that is by no means the rule, but that is just an example. The maximum that two women can ever be out of sync is 14 days. They would be at opposite ends of the cycle, and that is the maximum that they could be separate. On average, therefore, the onsets will be 7 days apart. on average, if we're taking the average of all the different points in the cycle that women could be, on average, the onsets will be 7 days apart. Given that menstruation lasts 5 days, on average, it's not surprising that friends commonly experience overlapping menstruation. And that's taken as confirmation of this synchronization theory. But that's going to occur by chance. And that wasn't accounted for in McClintock's study. So she used really poor statistical analysis and didn't account for this. Yes, there is going to be synchronization of menstruation, but it's not due to exogenous factors. It is simply just due to statistics and chance. So we need to consider that there are issues with this study. And therefore, it's not surprising that this finding, the McClintock effect, has failed replication attempts time and time again. So please don't use McClintock's research as supportive evidence for exogenous factors on infradian rhythms without discussing the methodology issues. However, it's not all bad. You know, there are some strengths of this. There is real-world application. When we're looking at the treatment of seasonal affective disorder, our understanding of infradian rhythms and our understanding of the effects of daylight and nighttime on the production of melatonin and serotonin has led to light therapy, which is a really effective therapy for people with SAD. It reduces seasonal affective disorder in about 80% of people, and it is preferred over SSRIs. It's deemed to be somewhat safer. It's deemed to be cheaper in the long run. The fact that light therapy basically stimulates strong light in the morning. So somebody will sit in front of a light machine, and that is there to reduce the release of melatonin, which will then suppress the release of serotonin. So it has had real-world application in improving lives for people based on our understanding of infradian rhythms. Okay, let's move on to look at ultradian rhythms. Ultradian, these have a frequency of more than one cycle in 24 hours, i.e. the cycle lasts less than a day. We have many in a 24-hour period. We may have many ultradian rhythms. The most researched ultradian rhythm is the stages of sleep, or the sleep cycle. Do not get this confused with the sleep-wake cycle, which is a circadian rhythm. This lasts 24 hours because it involves periods of sleep and wake. This is the sleep cycle. This is what occurs while you are sleeping. There are 5 distinct stages of sleep, and that lasts 90 minutes. Therefore, we have more than one cycle of 90 minutes in a day. Therefore, this is an ultradian rhythm. It is characterized by different brainwave activity measured by EEG, but we can also measure this through EMG, electromyogram, looking at muscles and how tense or relaxed they are, EOG, electrooculogram, looking at eye movements, and ECG, looking at heart rate, which all tell us information about those distinct stages of sleep. So let's just take a look at the overall sleep cycle. We can see here that we have a sleep cycle here, and here, and here, and here. So we typically have sort of 4 to 5 of these cycles in an evening. And we can see here the really distinct pattern of those stages. So we go from being awake to stage 1, 2, and then deep sleep 3 and 4, and then back up through stages 3, 2, and rather than waking, we then enter REM sleep, and that marks the end of our first cycle. And this continues throughout the lifespan. Throughout the nighttime. As I said earlier, we can distinguish between the different stages of sleep by looking at EEG recordings. Here we're looking at patterns of brainwave activity that are associated with different states of consciousness. And we can see that here we have alpha, theta, delta waves, and being awake is associated with beta waves. We can tell the difference between these different brainwaves by looking at their frequency and amplitude. Frequency is how fast they're going, and amplitude is how high they are. So if we look here, we can see that these are going really, really fast. They're not particularly tall, but we see many of them. So we say that this has got high frequency and low amplitude. If we compare that to what we see in delta waves in deep sleep, we can see here that they're slow, they're low frequency, and we can see that they're really high. So they're high amplitude. And this is how we can tell the different stages of sleep apart, by reading almost in real time the brainwave patterns that somebody is showing. So let's just go through those sleep stages. You don't need to know too much about it, no more than what's on here, but you might be interested and feel free to ask any questions in class. So stages 1 and 2, we call this light sleep. Somebody is easily awoken in these stages. What we see on the EEG is a decrease in alpha waves and an increase in theta-like waves. In stage 2 in particular, we also see these two phenomena: sleep spindles, which is sort of a burst of high frequency, low amplitude waves, and we see K complexes, a burst of high amplitude, low frequency waves. This tells us that somebody has now entered sleep. Then we enter stages 3 and 4. This is now deep sleep, referred to as slow wave sleep. Here we start seeing on the EEG recording lower amplitude, slower and higher, larger amplitude and lower frequency. So they're much slower and much higher waves. This tells us that somebody is now entering that deep restorative sleep. Dreams can occur, but they're less common in this stage. And this is really deep sleep. Somebody is very hard to wake up in these stages. If they do wake up, they're going to be feeling really groggy at this point. As I said, we can measure these stages through other measures other than EEG. And what we typically also then see is that things like heart rate, respiration, blood pressure is all decreasing. Somebody is in a very relaxed, very deep sleep state. Then we enter REM sleep. So we go from stages 1 and 2, 3 to 4, back up to 1 and 2, and then we enter REM. This is also referred to as stage 5 sleep by some. REM stands for rapid eye movement sleep, because on an EOG, an electrooculogram, we can see that the eyeball is moving rapidly back and forth. You might see this in a pet dog. If you see them dreaming, you can see their eyes flicking. The same happens to us. Our EEG shows beta-like waves. These are the same brain waves that we typically see when we're awake in a normal waking consciousness. So our brain is incredibly active at this point, but our body is almost paralyzed. Our heart rate is increasing, but sleep paralysis begins. We are not able to really control. We lose kind of all muscle tension, and we don't have any control in our bodies. So this is called paradoxical sleep, because we really see the difference here between our highly active brain and a really relaxed body. If we wake somebody from REM sleep, they will typically report that they're dreaming. This is when dreams occur, typically. Okay. The amount of REM sleep also increases across the night span. So we typically see longer periods of REM. Oh, it's annoying. So we can see that REM, you can't see it particularly well on here actually, but these periods of REM get longer and longer, which is why you always feel like you're dreaming in the morning, because you're just more likely to be in REM sleep, because we no longer have those stages of deep sleep. All right, so let's evaluate. Let's look at AO3 for ultradian rhythms. So most research into ultradian rhythms has been conducted in sleep labs. Sleep labs are really interesting places. Hard to get into. I used to do some school trips to sleep labs, and they were really cool, but unfortunately it's not possible for us to do that here because they're just too far away. But there is supportive evidence for the distinct stages of sleep in the sleep cycle. Dement is really the key researcher here. He is the pioneer of sleep research, and he started the first sleep lab. They conducted EEG recordings of participants as they were just having a night's sleep in the sleep lab. Based on their EEG recording, they then woke participants when they were displaying different types of brain waves, and they asked them to report their experiences. When they woke them during REM sleep, when they were displaying beta-like waves, they found that 80% had dreams. When woken in non-REM sleep, this is stages 1 to 4, only 20% of the time they reported dreaming. So this suggests that REM sleep is just an important part of this cycle, and it means that we can identify it through EEG recordings. The real benefit of conducting sleep lab research is that it is just highly controlled. It's not done in the person's own home. It is done in a lab environment where it's almost like a hospital room, actually. You're given a little bit more of a comfortable bed. There might be, some books and things in there to make you feel a little bit homey. There'll be typically a two-way mirror so people can observe you while you're sleeping, which can be a little bit disconcerting. But things like noise, temperature, distractions, they're totally soundproof rooms. All of that is going to be really, really controlled. We can also use EEG as well as lots of other recording devices. Electrooculogram, electrocardiogram, electromyogram. And if we're talking about just EEG alone, it has excellent temporal resolution. Now think back to your ways of investigating the brain. This means that we get almost instantaneous recordings of brainwaves. They're almost in real time, which means that we can, once we identify that unique pattern, immediately wake the person. And so we can get a really good sense of exactly what was the content of their dreams or whether they indeed even were dreaming at the moment that it's happening. However, sleep labs involve really complicated machinery, and it means that this highly artificial environment means that the sleep that we're recording and the information that we're gathering on ultradian rhythms may not represent ordinary sleep activity. You're in a different bed, you're wearing an uncomfortable EEG cap with all of those electrodes and the gel in your hair, you know that you're being watched by somebody, and all of this means that, typically we're studying people that have sleep issues anyway, and now we're putting them in a really unfamiliar environment. We're likely to see patterns of sleep that are not like their ordinary sleep activity. So it's really, really difficult for us to make generalizations about findings from research in sleep labs because it just might not be possible to capture ordinary sleep. The other thing that we just need to note is that there are individual differences in sleep. If we just have a look here at sleep across the lifespan, you can see some really interesting things about the proportion of non-REM and REM sleep, and also how much time that we are awake. So we can see that the amount of REM sleep decreases across the lifespan, as does non-REM sleep. Older people just get less sleep than newborn babies, for example. And so there are individual differences in terms of age across the lifespan. But there are also other individual differences in terms of the amount of time people spend, particularly in stages 3 and 4 sleep. And this is likely to be biologically determined. So although we're trying to describe ultradian rhythms and although we're trying to describe normal sleep, it may just not be possible to do in any meaningful way because there are just such significant variation between people.